Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute and the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hi, everyone. Welcome to the Genetics Podcast. We are bringing you a great episode from the archives this week that, based on our stats, probably the vast majority of you haven't heard yet, and we really want you to hear. The reason we picked this episode with Dr. Eric Green from Trace Neuroscience is they just had an amazing milestone where they have dosed their very first patient with a very innovative genetic medicine focused on UNC13A, which is a really interesting discovery that Eric talks through in the podcast that has major implications for ALS and potentially other neurodegenerative diseases. So their treatment targets this relatively novel pathway that a number of different groups are going after now. And I think it gives a really great window as well into genomics-driven drug discovery more broadly. And also Eric talks through his transition from academic clinical cardiology into co-founding two different major genetic medicine companies, Maze Therapeutics and Trace Neuroscience. So I really hope you enjoy this episode, and congratulations to the Trace Neuroscience team. Thanks, everyone, for joining us for another episode of the Genetics Podcast. I'm excited to be here with Dr. Eric Green, who's the CEO and founder of Trace Neuroscience, which is a biotech company focused on genomic medicine for people living with neurodegenerative diseases. Prior to that, he was co-founder and chief scientific officer of Maze Therapeutics. We're going to talk a little bit about the relationship between Maze and Trace and the interesting backstory there. And Eric's also an adjunct clinical assistant professor at Stanford. So definitely a busy man. We're glad to get you here and have some of your time to talk about your work. I'd love to start with actually a company I didn't mention, which is MyoKardia. This was your first foray into biotech and precision medicine. I'd love to just hear a little bit about your career arc more generally, how you got into genomics and precision medicine, and we'll start there and work our way forward to where you are today.
Thanks, Patrick, and appreciate the introduction. Very glad to be here. Briefly by background about me, I'm a cardiologist and physician scientist by background. And when I was in academic cardiology, this was a time where we were starting to see really a huge impact of genetics and the genomics revolution in areas like oncology, but it really hadn't started to make an impact in cardiology. And so one of the big questions that I spent my time thinking about in academics was how could we bring some of the impact of the genomic revolution to cardiology? And one of the areas where we really had an understanding of the genetic drivers of disease was in cardiomyopathies. And this is a set of diseases characterized by dysfunction of the heart muscle, and where through mapping in families and linkage had come to understand the genetic drivers of those diseases in the 1990s. And these were mutations in the sarcomere motor proteins of the heart. But despite that genetic understanding, we really hadn't been able to leverage that into developing new therapies. And so that was the fundamental underlying question that we faced when we started the company MyoKardia, which is how could we take advantage of our genetic understanding of those diseases and use it to develop new medicines that could really improve the lives of patients with heart failure.
What was the blocker that was making it difficult to translate those genetic insights into therapeutics at the time?
Yeah, and I think as we go through the discussion, I think in a lot of ways understanding what the limitations were there, I think have been a roadmap for me as I think about how we go from translating genetics into medicines more broadly, and that is. It was the mid-1990s. We had, as a field, defined the genetic drivers of a disease, but it was not at all clear how those proteins were actually working in leading to cause disease. And then in addition, how could you think about actually modulating them? So this really set off several decades worth of really detailed mechanistic work. And Jim Spudich, who's a professor at Stanford, had spent several decades, even before this genetics was defined, trying to understand how myosin motor proteins worked across all organisms, down from single-celled organisms all the way through to humans. And what I think really made myocardia possible was bringing together that underlying genetic understanding of disease with really deep understanding of the mechanism by which these proteins were working. And that was the original marriage that really was at the start of myocardia. And those two things, and then I think the third really big advance came actually from another biotechnology company, a company called Cytokinetics, that had discovered that you could find small molecules that could actually bind directly to these motor proteins of the heart and modulate their function. And once we had those three pieces in place, which is really strong conviction in the target based on genetics, detailed molecular level mechanistic understanding of how those proteins were working in disease, and then an understanding of what is a modality and a mechanism by which you could actually intervene on those proteins. Once those three pieces were in place, that really was what made myocardia possible. And we set out to identify small molecules that could bind to the sarcomere motor proteins of the heart and turn down their function, with the idea that this could be an effective treatment for people with hypertrophic cardiomyopathy whose disease was caused by mutations that led to overactive motor proteins in the heart.
And if my memory is correct, myocardia was founded initially by Third Rock Ventures, which I'd love for you to tell people a little bit about. Third Rock Ventures, a very very famous and successful venture fund, but also that more than investing in companies, they incubate companies from very early scientific stages. After you left myocardia, I think you went there and we're working on Maze Therapeutics behind the scene. Maybe you can tell people a little bit about Third Rock Ventures, how the process for incubating and spinning out companies works, and maybe through the lens of your founding of Maze Therapeutics, which I think happened next.
Yeah, so Third Rock Ventures is a venture creation firm that was started back, I think, in 2007, with what at the time was a really unconventional approach to starting and investing in biotech companies, which was that they weren't going to sit in a conference room and wait for entrepreneurs and companies to come to them for investments. They were going to identify areas of emerging science and medicine where they thought there was an opportunity to build companies. And then they would bring together both academic founders as well as biotech executives to form companies in those areas. And then, like more traditional venture firms, would also provide investment in those companies, as well as a lot of support and advice in the construction and building of those companies. And one of the companies that Third Rock got involved in building now back in about 2011 was myocardia. And it really was based on seeing those confluence of factors that we talked about, which is this is an area where we hadn't really seen a big impact of genetics, but had some of that underlying foundation in genetic understanding. And so brought together both the concepts, but also really the key academic leaders. So John and Cricket Seidman, who had made the fundamental genetics discoveries, Jim Spudich, who had done so much work on the mechanism, as well as Leslie Leinwand from the University of Colorado, who had also done a lot of work to understand the cell biology of those myosin motor proteins, and really played a pivotal role in the underlying concepts, as well as aggregating the people and the financing to make that company possible. And that now has, I think, extended to over 60 different companies that have largely been built on that model, which is really starting from a blank sheet of paper of what are areas where we think there's an opportunity to apply cutting edge science to really important areas of medical need. And when I was sort of looking at the landscape as I was thinking about moving from academia into biotech, it just was an approach and a group of people that really resonated with me, who saw biotech companies really as an opportunity to translate some of the exciting discoveries that were coming out of all the fields of biology and genetics, and really think about how can we turn these into medicines in the most efficient way possible.
What had you thinking about transitioning from academia into biotech? We're going a little out of chronological order, obviously, but I'm curious to hear about that.
Yeah. I think I first had the notion that I wanted to be in the world of biotechnology and entrepreneurship when I was a graduate student. I was an MD PhD student at Stanford, where in addition to developing my expertise in medicine and basic science, got really interested in entrepreneurship and in the way that I think many people who spend time in the Bay Area, especially in those days in the early 2000s, did, and got involved in building a few companies out of work that I had done in a course at Stanford. So took a course through the Stanford Design School where they put together interdisciplinary teams to work on challenging problems in the developing world. And we took a trip actually out to um, health clinics in Mexico with really very little direction. Just said, go spend some time here,
Mm.
live with the folks who are taking care of patients in these rural clinics, and identify important needs that they have, and then use that as a springboard to start thinking about solutions. And we ultimately developed a very low-cost device for treating asthma in those patients that went on to win the Stanford Entrepreneurship Challenge. And I think that for me
Wow.
was in a lot of ways the out of the birth of my recognition that I loved being in that entrepreneurial world, bringing together people with different backgrounds, all of whom were really committed to solving an important problem and just laser focused on it day and night. And I wanted to eventually come back to that environment, but to do so from a place of stronger grounding in science and medicine. And so went on to continue my training in medicine and in cardiology, but was always looking for what was going to be the right place, the right environment to come back to that entrepreneurial path. And that was when I found Third Rock, and I think it really was a great combination.
Yeah, so 2017 and 2018, you were in Third Rock, basically building what would become Maze in stealth. February 2019, you all launched with 100, I think 190 million in funding, and a focus on using genetics and genetic modifiers to discover new drugs. And this, there had been discussion, I think, for a number of years on the importance of genetic targets in drug discovery, but it was still back then not consensus that certainly biotech should be basing their entire pipeline on something like this. So that was, I guess, the non-consensus and right bet that you all were making at the time. Tell me a little bit about that. How much did the initial concept drift during that time? And for those who aren't familiar with Maze and what's been happening over the last few years with the company, how it's been.
Yeah. So, just as you say, in that period of time around 2017, I remember this very well, that this was the time where the UK Biobank was finally going to be releasing its first genetics data. And I remember having the distinct sense that it was sort of like we were living on the frontier in the West, that there were all these groups that sort of had their computers queued up to say, okay, we're now going to be able to do, the GWAS, the largest GWAS of almost every trait.
Yes.
And it was an exciting time to be talking to folks in academia about this, but it also got me thinking, well, what is this going to mean from a drug discovery and biotech standpoint? We at this point had made a lot of progress at myocardia, had taken our lead drugs into the clinic, demonstrated proof of concept, and so I had a really strong conviction that basing drug discovery programs in biotech companies on a foundation of genetics was a really powerful way to go. And now we had the opportunity to think about doing that, not, on one target in a really deep way, but how could we think about doing this really at scale? And in some ways felt like this was kind of a challenge to us as an industry, which is, we had made phenomenal progress at myocardia, but this was now probably 25 years after the initial genetics discovery was made. And, with us now having thousands, tens of thousands of new genetic insights coming online, how were we going to make that process more efficient and more scalable? And that was, I think, the challenge that really led me to leave myocardia, come back to Third Rock, and start working on Maze. And we had a couple of, I think, key, what I call key theses when we were building Maze, many of which were around genetics. And one of them was that there had been a fundamental shift in the last at that time where if you wanted to apply genetics in drug discovery up until then, and there were some great companies that had made that commitment even quite a bit earlier than that. Step one for them was to build a massive biobank. And you're talking here about companies like Regeneron, companies like Decode, who had, I think, really been very prescient in doing that. But that was a huge, time-consuming and expensive commitment. And with the advent of the UK Biobank and some of the other global biobanks that were coming online, one of our key theses was, the critical expertise was no longer about actually building and aggregating those genetics data, but about thinking about how to analyze and work across those data sets, and that lent itself much better to the kind of work that could be done in a focused biotech company than saying, yes, we need to somehow take 1 million samples and sequence them all and then figure out how to actually do the underlying analysis. And so that was, I think, a key insight that we had that really made that the right time to think about building a company that was going to, across multiple different areas, think about how they were going to translate insights from genetics into medicines. I'll tell you about another thesis that we had at the start of that company, which I think proved to be less sort of well-timed, which was that we were at a moment where we were seeing a real growth in different therapeutic modalities as
Yeah.
well. This was both small molecules and antibodies, but we were now starting to see a lot of exciting data coming out of particularly genetic therapies, like gene therapies, like oligonucleotides. And the one of the challenges of building a genetics first company is that you don't necessarily know what's going to come out the other end. You think you're going to have some confidence in the targets that are going to come out, but by nature they may be in different therapeutic areas and they may lend themselves well to different modalities. And I think our thesis at the time was that we were entering an era where those access to those modalities and expertise in those modalities was becoming more commoditized. And I think that, well, I still ultimately believe that thesis is going to be proven true. I think we were a little bit early on that, and we'll come back to this because I think in a lot of ways this informed the approach that we took when we had the insights that led us to Trace, and what motivated us to say that this really is going to be better suited to as a standalone company.
Yeah, I love that you brought that up because I think it's a thought I've had a number of times. If you're investing in large scale genetics efforts and you're someone like Regeneron that can do small molecules and do biologics and do gene therapies, then no matter what the genetics machine spits out, you have some confidence you can go for it. But that's a lot harder in a biotech. You can't necessarily afford a chemist and and a gene therapy team. So yeah, I'd love actually if you could talk a little bit about how Trace was born and how it fits into this broader thesis, because I'd love to come back and spend a little bit more time on that, because it is I think it's important for any biotech that's investing in genetics to think really carefully about how this affects the downstream and how they partner or how they figure out ways to choose the right therapeutic modality, regardless of what the science is telling them is the right thing to do, versus choosing to only do small molecules, for example, and constraining the space you can operate in.
Yeah, and this is, I think, one of the real things that I learned through our experience at Maze is that if you really are prioritizing genetics as the first place to go, almost inevitably you're going to end up in a range of therapeutic areas and a range of modalities. And from a kind of biotech company strategy standpoint, you need a point of view on how you're going to deal with that. And one point of view can be to say we're just going to exclude anything that doesn't fit into some narrow set of that 2x2, meaning
Right.
some therapeutic area and some modality. And in a lot of ways, for a traditional biotech, that's the most sensible approach. It's challenging to think about building biology expertise and translational medicine expertise across a number of therapeutic areas. And similarly, it's challenging to really build the expertise in multiple modalities, particularly if some of them are not traditional, more standard approaches. What I think we tried to do at Maze and had some success with was to be able to think more creatively about a business model that went beyond just exploiting that narrow set of indications and therapeutic modalities. And one of the examples of this was that we were able to find a number of really promising targets in ophthalmology that were really much better suited for antibodies than they were for small molecules, and ultimately took those programs, spun them out into a new company called Broadwing Bio. And that was, in a lot of ways, sort of the beginning of our thinking about how do we how do we take advantage of and find a way to move forward targets that we really like, but that don't fit into that area of the therapeutic area modality plot that is where we really have our expertise and we want to focus our attentions. And I think that was the backdrop for what happened with us as we came upon what was a really exciting discovery about the gene UNC13A in ALS. So maybe to back up a little bit, when we started Maze, we recognized that there was this really wide range of opportunities across disease areas, but that we were going to need to figure out, at least initially, where do we want to focus our attentions. And my priority was to say, where can we identify some areas where we think that the genetics resources are becoming mature, and if we could make this work, would just have some of the biggest impacts on patients. And very quickly, one of the areas that rose to the top of our list was ALS. We were finally starting to see the size of the genetics resources becoming mature. We were starting to see some real progress in developing therapies for rare familial forms of ALS that were driven by mutations, for example, SOD1. But in the vast majority of people with ALS, we really had no treatments that were rooted in genetics and that were making a meaningful impact on disease natural history. And this was, I had the opportunity to meet a number of people with ALS during my time as a physician. And, what I've found is that meeting any of those patients sticks with you. And that was certainly the case for me. And it had always been a disease that I'd said, if we can find a way to make a difference here, it would be transformational. And so from early on, ALS was one of the disease areas we had been interested in. And we started working with a faculty member at Stanford by the name of Aaron Gittler, who had also been thinking about how genetics could be useful in developing medicines and understanding the biology of ALS. Aaron started this in an era well before one could do this with human genetics, and so started actually thinking about yeast biology and how that could be applied to think about identifying new targets. And so we started working with Aaron on this question, and he originally had insights around a genetic modifier of ALS called ataxin-2. But we were continuing to think about how we could use genetics to identify new targets. And there's another theme that I'll bring up here that I think I've seen a number of times in my career also, which is that what really was unlocking here was not finding a new GWAS locus, but coming to a new understanding of how a genetic signal was working in the mechanism. And this was the case with UNC13A. So UNC13A had been identified as one of the first GWAS signals in ALS back in 2009. The two signals that came out of that screen are ones that are still, the most robust hits today, which are C9orf72 and UNC13A. And it had sat there for a little over a decade as a very robust genetic signal, but one for which we didn't understand how it was working in disease. And UNC13A is a very interesting genetics target because it came here, as we're talking about, out of modern genomics, but also has a history in very classical model organism genetics. So it was first identified in genetic screens in C. elegans by Sidney Brenner back in the 1960s, where large-scale mutagenesis was done in worms, looking for genes that, when mutated, led the worms to become uncoordinated. And so that's where the name UNC comes from, is from that phenotype that was observed in worms. Over the course of the next several decades, really detailed biochemical work was done that led to a Nobel Prize understanding what it was that this protein was doing, and finding that UNC13A is an essential component of the presynaptic vesicle release machinery that's necessary for the priming and docking of vesicles at the presynaptic terminals. And for that reason, in worms, but also true in rodents and in humans, if that protein is lost, those vesicles can't dock, and this results in paralysis. So this was a gene for which we had both a lot of classical genetics as well as this strong signal coming out of genomics. And yet, this was not enough to really enable therapeutics development because of that key missing gap around mechanism. And this is where the work that we did with Aaron really unlocked this target. And what we discovered was that UNC13A, the mRNA for UNC13A, under normal conditions is bound by a protein called TDP43. And TDP43 is a nucleic acid binding protein that in healthy people cycles back and forth between the nucleus and the cytoplasm. But in 97% of people with ALS, the protein gets stuck in the cytoplasm. And as a consequence of that, it's no longer able to bind the mRNA for UNC13A. When that happens, it unmasks a cryptic exon in UNC13A, and that cryptic exon gets included. And the consequence of that is that the mRNA is subject to nonsense-mediated decay, and the protein is deficient. And that insight really pointed a new direction towards understanding how it was that UNC13A was involved in ALS, which is that it's lost, but also what the mechanistic basis for that is, which is the inclusion of this cryptic exon.
Yeah.
And this now gave a very clear direction to how one could think about developing therapies here. And it took advantage of some recent advances in modalities, particularly with antisense oligonucleotides, where we'd seen in the example of spinal muscular atrophy in the development of the medicine nusinersen, that if you could find an antisense oligo that could bind directly to a region of the genome, in this case, or region of an mRNA, in this case, the cryptic exon, you could prevent its inclusion in the mature RNA. And so that was the therapeutic hypothesis that came out of the basic science work that we had done with Aaron. And one interesting link that I'll mention here, because I think particularly for geneticists, this is very satisfying, is that the genetic variants that had been associated with ALS in UNC13A, and I should say not only associated with the risk of developing disease, but with disease progression. And this was an important thing that made us very excited about this target. Those genetic variants sit right in the region of that cryptic exon that gets included when TDP43 is excluded. And so there's this very satisfying understanding that these genetic variants, which increase the risk and the rate of progression of disease, sit right in that region that is bound by TDP43. And when the risk variants are present, they interfere with the binding of TDP43. So we now had a very clear and compelling story about how these genetic variants were acting and how they were serving to exacerbate disease and what one could do to try and correct those effects. And so that was the insight that we had now back in the early days of COVID, that we made this discovery in 2020 and very quickly said, this is exciting biology, but this is also potentially really enabling from a therapeutics discovery standpoint. And it was at this point that we started to say, in a lot of ways, this is something that fits in well with what we're doing at Maze, very much motivated by genetics in the area of a disease of high unmet need. But we were fortunate at that time at Maze to be making a lot of progress in a number of different therapeutic areas. Had a program in rare disease that was going well in Pompeii. Had several programs in kidney disease, including one that I know you've talked about on the show here before, which is APOL1 mediated kidney disease. And as we thought about the opportunity here in ALS with UNC13A, in the context of all those other programs, it just became clear to me that if we were going to give it its greatest chance of making an impact on patients, we needed to put it in a different context, and one where we could combine all the expertise that we needed from a modality standpoint, because we've never done an antisense oligo at Maze, all the expertise in the clinical development of medicines for people with neurologic diseases, particularly this is going to be an intrathecal ASO, and so that has a lot of special expertise associated with it. And then, maybe most importantly, I think recognizing the benefit that comes from having a team with a really clear focus and whose first priority every day is, how are we going to take this medicine for people with ALS and make it a reality? And so I think all of those factors started to coalesce back from the time of that original discovery, and culminated earlier this year with us saying, hey, we're going to take the work and the team that we had started at Maze, which really was very enabling, but put it in a context where it's going to have a greater likelihood of success. And that's the company that became Trace Neuroscience that we launched just a few weeks ago.
Congratulations on the launch. It's exciting. And it's really interesting how that kind of fits into the thesis you laid out at the beginning. You've got Maze, which has generated a lot of different genetically driven programs, but it sounds like Broadwing is one example where you've found actually we need to go a biologic approach. Trace is an example where you needed to go an ASO approach. And so it kind of does validate the thesis in a way that you've just found a different way to do it, right? It's spinning out companies and having some relationship, I guess, to the to the original founding mothership, but needing to resource those in a dedicated way to build out the team, as you mentioned. So if you could tell people where are you timeline wise with Trace? How close are you to going into the clinic? Obviously, you've got a big head start because you all have been working on this at Maze for a little bit before you've spun out. But yeah, there's always a lot of work to do before you start to go into the clinic. So I'm curious about that.
Yeah, and there are a couple elements of the Trace story that I want to share here. One of which was that I told you about how Aaron Gittler and our team started working on this and made this key discovery. We learned not long after that almost simultaneously there was another group led by Pietro Ferrata at the University College London who came to that same insight. So as with so many of our key advances in science, it turned out that there were multiple groups that were really converging on this. And one other key piece of the Trace founding story was that we wanted to really bring together all of the key leaders and scientific discoverers into one company, which we thought would give us the greatest likelihood of success and just access to as much of the expertise as possible. And so I say that because I think a lot of the rapid progress we've been able to make at Trace is attributable to really having brought together that strong foundation. So as a consequence of that, we now have been able to identify antisense oligos that can very effectively suppress the inclusion of that cryptic exon. We've now taken them through preliminary toxicology studies across rodents and non-human primates and are really seeing a very attractive emerging safety profile. So we have a molecule that we think is ready to start advancing towards clinical development. The goal of getting that molecule into the clinic and into patients in the early part of 2026. The other big aspect of what we're doing now, and another place where we're really thinking about how we can continue to leverage genetics, is in trying to design the most efficient and informative clinical development program, and particularly in the area of biomarkers. And this is a place where I think we, as a field, have started to be able to really apply some of the insights coming out of biobanks, but that we're still really in its infancy. And I told you about these genetic variants in UNC13A that are known to be associated with disease risk and disease progression. And that fact alone has been really valuable for us in setting the foundation for the program. But we're looking to continue as much as we can to leverage those genetic variants to help us get a better understanding of what might be good biomarkers for our therapy, because the idea is that these genetic variants exacerbate the splicing of UNC13A, and that we're developing a therapy that we expect is going to have the opposite effect to suppress the missplicing. And so we're able to take advantage of large data sets where we have people who carry these genetic variants. We also have now growing bodies of multi-omics information about circulating biomarkers, whether they be protein biomarkers, metabolite biomarkers, and other things, to really give us direction for how we think about what might be the most effective markers to track that will show us that our therapy is having the intended pharmacological effects. And so this is, for me, a really exciting frontier in how we can apply biobanks in drug development, where I think we have a unique opportunity here, in part because of how common our genetic variants are, that we're able to have access to large numbers of people who both carry these variants, and where we have the kind of detailed phenotypic and multi-omic information to do discovery.
One interesting thing about this that I want to come back to is the odds ratio of the gene is not huge, and I think this is an important one to come back to, because sometimes people equate big odds ratio, great target, but this isn't one of those cases, right? It's 1.2, 1.3. Maybe you could talk a little bit about that and how you knew not to ignore this relatively low odds ratio gene and hone in on the importance.
Yeah, and I remember very well. So, one of the academic founders at Maze was Mark Daly, and I remember very early on in our days of building Maze, we were thinking about, what in our mind made a good genetic target. Hearing a talk from Mark where he gave the example of HMG-CoA reductase in LDL and said, well, common variants in HMG-CoA reductase lead to very low odds ratios for changes in LDL. Statins, which are molecules that go after HMG-CoA reductase, have become the most successful,
Yeah.
one of the most successful classes of medicines ever, and can have enormous effects on LDL levels in people. And I think that one example has always stuck with
Hmm.
me as an illustration that, effect size and odds ratio not always equal to pharmacologic effects of intervening on a target.
Why is that? Is that just because the super large effects are rare in nature, or why is that?
Well, so I think there's a couple aspects to it. One is that naturally these more common variants in general tend to have smaller effect size. Now there's some exceptions to that. APOL1, which we can
Yeah,
talk
it's a good
about,
example.
is an exception, and in fact, I think the exceptions can often be very fruitful places to look for drug targets because you get a lot of ways the best of both worlds, which
Yeah,
is a common variant that's relatively easier to find, also easier to find people to study, and conviction in the effect size of the
right.
intervention. But in many cases, these common small effect size variants can be very informative for drug discovery, provided that the variant itself is having a relatively small impact on protein function, and you then can attempt to have a pharmacologic intervention that can modulate that target to a much greater extent. And that's exactly what the case is with HMG-CoA reductase. And I think there's a similar pattern that we see with UNC13A, which is that these risk variants in UNC13A exacerbate the missplicing of the mRNA, but to a relatively modest degree. And we with our antisense oligos are able to entirely correct the missplicing of UNC13A, and so have that same hypothesis that we talked about with the statins, where we imagine that the pharmacologic effect here can be much
Yeah.
greater than the genetic effect. And there's even an additional layer, based, I think, on what we know about rare variants in UNC13A. So I talked about rare, damaging variants and what they can do in worms. We actually are part of a publication that's going to be coming out in early 2025 on rare variants in UNC13A in humans. And
Mm.
much as was described in worms, rare, highly damaging mutations in UNC13A can lead to very severe phenotypes in humans as well. So, people who lack nearly all UNC13A from conception have a very severe and early onset neurodevelopmental
Oh,
and
wow.
neuromuscular disease. And I think that uh, kind of expansion of the allelic series here gives us even
Yeah.
greater confidence that there is really a lot of room to go in terms of correcting UNC13A dependent phenotypes in humans.
Yeah, that's fascinating. You mentioned APOL1 as well. I just wanted to let people know if you go all the way back to episode 116, we had a great discussion where we went deep into this with Alex Bick, who I know you know well and work closely with. I'd love to just hear your perspective on that story as well. That's probably the lead program for Maze right now, right? And it falls in this category that you mentioned of great target, major effect size, relatively common, especially in people with African ancestry. I'd love to just hear a little bit of the story behind that because it's there's a couple of companies going after that target right now, and it's an opportunity to have a really big impact, I think, for the people who figure it out.
Yeah, absolutely. And as a clinician, this observation that African Americans have a greater burden and greater severity of kidney disease is one that is, just ever present in the care of patients, particularly as a cardiologist, something we see all the time. And when it became clear that one important driver of this disparity was this underlying genetic difference in APOL1, it really motivated me to think about how can we now take that genetic insight and turn it into a medicine. And much like the example we talked about in myosin, much like the example we talked about in UNC13A, the genetic insight long predated the development of medicines here. So it was a decade before people started to think about, well, how can we actually build on this genetic insight? And the big reason why is that there were variants that had been identified in APOL1, very clearly linked to the development of chronic kidney disease, end-stage renal disease, more rapid decline in renal function. But there was not a clear understanding of how it was that those variants were working. And people had a lot of different theories about how it was working and therefore what one might want to try to do therapeutically. APOL1 circulates as a protein. So there were approaches to think about, well, can we bind that circulating APOL1 with antibodies? Is that going to be the way to go? And so not only did people not know the function, it wasn't even clear what the
Mm.
appropriate tissue was to be going after this protein. And this was where we really tried to focus our genetics attention, not on discovering the target, but on thinking about, can we use genetics to give us greater insight into the mechanism? And in this case, this didn't come from those disease-driving variants, the APOL1, G1, and G2 variants. It came from another very rare variant that had been described in a case report. In fact, a case report that was related to an entirely different function of APOL1. So we're talking here mostly about APOL1 and its role in kidney disease. The first discovered role for APOL1 was as a circulating factor that could prevent against trypanosomal infection. And we had been very intrigued to see a case report of somebody who developed very rare trypanosomal infection and who was found to have an inactivating mutation in APOL1. And so the question that we asked together with Aaron, with Alex, is, might that same variant that eliminates protection against trypanosomes, might it also modulate the risk in kidney disease? And that was the key insight that we had together with Alex, taking advantage of the Million Veterans Program data set, which was really a unique data set to answer that question. And then what the work our laboratory did in tandem was to say, if that variant is protective against kidney disease, which is what we found with Alex, how is that variant working? And we found that the way that variant functioned was to reduce the poor function of APOL1. And that really gave us the clear conviction that the way to therapeutically approach this protein was to mimic the effects of that protective variant and block the pore of APOL1. And so it's a nice example of how sometimes the key application of genetics really doesn't have to do with identifying the target in this case, but was really about getting conviction in the mechanism and then giving us direction for how we were going to conduct our drug discovery campaign.
Yeah, that's really interesting, and it also, I think, supports one of the questions I've heard a lot of people in genetics and small biotechs ask is, what can we do when some of the large players have the kind of resources they have to throw at sequencing huge populations and understanding and maybe being the first to find new targets, right? If you've got access like Regeneron does to millions of exomes, they're probably going to be the first to discover new targets. But to the point that you just made, it doesn't mean the job is not done from there. It often takes years of work to figure out actually how do we understand, given the new target or a target that's been around for a long time, how do we actually best treat it? And it sounds like genetics has an important role to play there. And also there are other questions like, is it safe to knock out, right? That these large population biobanks can be really, really helpful with from a PheWAS perspective. That's really interesting. To me, it gives some hope to the mid-sized biotechs that do want to make genetics a core part of what they do.
Yeah, and I think it's a very important point, and one I believe in strongly. It's very much the exception rather than the rule that the genetic discovery itself makes the therapeutic approach self-evident.
Yeah.
Wish that were the case more often, but I think it's very rarely the case, and that what it takes in most of these cases is careful attention to the genetics, not only the first variant that's discovered, but really trying to elaborate allelic series in these genes, and then combining that with in-depth biology to really understand how those variants are working, and that those two steps, in a lot of ways benefit from context in a company that can give deep focus to one or a small number of these genetic insights and think about how they can be turned into medicines.
Amazing. Eric, thank you. I know we're running out of time here. If people want to follow your work, I know they can go to traceneuro.com, and you've got a beautiful website that describes what you do really very well. Is there anything in particular you're looking for? Are you hiring? Are you looking for partners? We could take this opportunity to call it out.
Yeah, I think we're always looking for partners, both on the side of people who want to work with us as we move this program into the clinic. We think this is a really exciting and differentiated opportunity to bring the first real genetically rooted therapy to the broad population of people with ALS. And then, as we talked about earlier, we're very much looking to leverage genetics to help inform how we do that development. And this relates to biobanks where we may be able to relate the effects of variants in UNC13A to different, whether it be molecular, imaging, clinical phenotypes, really to help us in thinking about what are going to be the most informative markers to follow. So those are collaborations we're very interested in, but also areas that I look forward to updating you and your listeners in the future.
Amazing. Well, congratulations, and thanks again for taking the time to be with us. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.